I'm going to deliver a lecture on simple single factor inheritance in men. One of the most remarkable characteristics of chromosomes is the ability to sort precisely the genetic material represented in homologous pairs of chromosomes into daughter cells and gametes. This assortment is recognized through the many visible characteristics of individuals. This phenotype or visible presentation of a person is influenced by the expression of alleles at different times during development, at different efficiencies, and in different cells or tissues. Observed differences are the result of a cell's genotype or molecular variation in alleles. Mechanisms of inheritance generally refer to traits resulting from a single factor or gene called unifactorial inheritance, or from the interaction of multiple factors or genes called multifactorial inheritance. Because it is the simplest inheritance pattern, unifactorial inheritance is the best understood. Gregor Mendel first investigated this type of inheritance in his famous studies of garden peas in 1865. Because the underlying principles of Mendel's work became hallmarks to understanding inheritance. Mechanisms of unifactorial inheritance are often called Mendelian inheritance, and the other mechanisms are referred to as non-Mendelian inheritance. Single factor inheritance in man is the derived conclusion for Mendelian genetics. Although there are various ways of genetic variance in human, the basic model starts from Mendel's one-way transmission, in which the two alleles of a particular organism plays a distinctive role in deciding about the genetic variation and form of the organism. Inheritance patterns trace the transmission of genetically encoded traits, conditions, or diseases to offspring. There are several modes of inheritance. Number 1, single gene inheritance or Mendel inheritance. Number 2, multifactorial, and number 3, mitochondrial. The present paper will discuss on single gene inheritance. Genes are found on autosomes and sex chromosomes, and evidence for the existence of genes prior to the molecular revolution was based on measurable changes in phenotype. These changes resulted from allelic variation. Observing variation depends on the relationship of one allele to another. The terms used to describe this relationship are dominant and recessive. If only one allele of a pair is required to manifest a phenotype, the allele is dominant. If both alleles must be the same for a particular phenotypic expression, the allele is recessive. This is described by the notation capital AA, capital A, small a and small a, where capital A is dominant and small a is recessive. The capital AA condition is called homozygous dominant. Capital A, small a is called heterozygous, and small a is called homozygous recessive. Genetic conditions caused by a mutation in a single gene follow predictable patterns of inheritance within families. Single gene inheritance is also referred to as Mendelian inheritance, as they follow transmission patterns he observed in his research on peas. There are 4 types of Mendelian inheritance patterns. Number 1, autosomal dominant. Number 2, autosomal recessive. Number 3, X-linked dominant. And number 4, X-linked recessive. Dominant conditions are expressed in individuals who have just one copy of the mutant allele. Affected males and females have an equal probability of passing on the trait to offsprings. Affected individuals have one normal copy of the gene and one mutant copy of the gene. Thus, its offspring has a 50% chance of inheriting the mutant allele. There are numerous examples in humans of defective genes that are transmitted in a dominant pattern. Achondroplasia, a form of dwarfism, is inherited as an autosomal dominant trait. Achondroplasia is a congenital disorder, a defect present at birth. Affected individuals are small and disproportionate, with particularly short arms and legs. With an estimated frequency of 1 in 15,000 to 40,000 live births, achondroplasia is one of the more common Mendelian disorders. Most infants affected by achondroplasia with 2 mutant alleles representing a homozygous condition are stillborn or die in infancy. Heterozygous individuals surviving to adulthood produce fewer offsprings than normal. This observation underscores an important point for many autosomal dominant disorders. 2 mutated alleles often have severe clinical consequences. Characteristics of autosomal dominant inheritance. Guidelines for recognizing autosomal dominant inheritance in humans may be summarized as follows. Number 1. The affected offspring has one affected parent unless the gene for the abnormal effect was the result of a new mutation. Number 2. Unaffected persons do not transmit the trait to their children. Number 3. Males and females are equally likely to have or to transmit the trait to males and females. Number 4. The trait is expected in every generation. Number 5. The presence of 2 mutant alleles generally presents with a more severe phenotype. Detrimental dominant traits are rarely observed in the homozygous state. Approximately half of the children of affected parents inherit the condition and half do not. Examples of autosomal dominant diseases are Huntington disease, achondroplasia, short limb dwarfism, and polycystic kidney disease, etc. Huntington's disease, a progressive neurodegenerative disorder, is a well-known example of an autosomal dominant single gene disease. Most individuals with a single copy of the mutant Huntington gene HTT will have Huntington's disease later in life. Typically, autosomal dominant diseases affect the individuals in their early years and prevent them from living past infancy or childhood, which in turn precludes these individuals from reproducing and potentially passing on the mutation to their offspring. In the case of Huntington's disease, however, the late onset of the disorder means that many affected individuals have already had children before they are even aware that they carry the mutation. Autosomal recessive. Recessive conditions are clinically manifest only when an individual has two copies of the mutant allele. When just one copy of the mutant allele is present, an individual is a carrier of the mutation, but does not develop the condition. Females and males are affected equally by traits transmitted by autosomal recessive inheritance. When two carriers mate, each child has a 25% chance of being homozygous wild type means unaffected, a 25% chance of being homozygous mutant means affected, or a 50% chance of being heterozygous unaffected carrier. The inheritance of such recessively determined traits in humans are albinism, cystic fibrosis, and phenylketonuria. Special significance is attached to the heterozygous carrier, the individual who unknowingly carries the recessive allele. It is usually difficult to tell prior to marriage whether the individual bears a detrimental recessive allele. Thus, a recessive allele may be transmitted without any outward manifestation for several generations, continually being sheltered by the dominant normal allele. The recessive allele, however, becomes exposed when two carrier parents happen to mate. This explains cases in which a trait absent for many generations can suddenly appear without warning. Often, only one member in a family is afflicted with a particular disorder. In such an event, it would be an error to jump to the conclusion that the abnormality is not genetic solely because there are no other cases in the family. Without a positive family history and sometimes the corroboration of diagnosis, the occurrence of a single afflicted individual may represent a new sporadic mutation. Characteristics of autosomal recessive inheritance. Guidelines for recognizing autosomal recessive inheritance may be summarized as follows. Number 1. Most affected individuals are children of phenotypically normal parents. Number 2. Often more than one child in a large subset is affected. On average, 1/4 of siblings are affected. Number 3. Males and females are equally likely to be affected. Number 4. Affected persons who marry normal persons tend to have phenotypically normal children. The probability is greater of marrying a normal homozygote than a heterozygote. Number 5. When a trait is exceedingly rare, the responsible allele is most likely recessive if there is an undue proportion of marriages of close relatives among the parents of the affected offspring. Affected individuals are indicated by solid plaque symbols and unaffected carriers are indicated by the half plaque symbols. Examples of autosomal recessive diseases. Several human diseases including cystic fibrosis, sickle cell anemia, and oculocutaneous albinism also exhibit an autosomal recessive inheritance pattern. Cystic fibrosis is associated with recessive mutations in the CFTR gene, whereas sickle cell anemia is associated with recessive mutations in the beta hemoglobin HBB gene. Interestingly, although individuals homozygous for the mutant HBB gene suffer from sickle cell anemia, heterozygous carriers are resistant to malaria. This fact explains the higher frequency of sickle cell anemia in today's African Americans who are descendants of a group that had an advantage against endemic malaria if they carried HBV mutations. Finally, oculocutaneous albinism is associated with autosomal recessive mutations in the OCA2 gene. This gene is involved in biosynthesis of the pigments melanin, which gives color to a person's hair, skin, and eyes. X-linked recessive. X-linked recessive traits are not clinically manifest when there is a normal copy of the gene. All X-linked recessive traits are fully evident in males because they only have one copy of the X chromosome, thus do not have a normal copy of the gene to compensate for the mutant copy. For that same reason, women are really affected by X-linked recessive diseases. However, They are affected when they have two copies of the mutant allele. Because the gene is on the X chromosome, there is no father to son transmission, but there is father to daughter and mother to daughter and son transmission. If a man is affected with an X-linked recessive condition, all his daughter will inherit one copy of the mutant allele from him. X-linked inheritance is distinguished by the presence of one chromosome in males but two in females. Guidelines for recognizing X-linked recessive inheritance may be summarized as follows. Number 1. Unaffected males do not transmit the disorder. Number 2. All the daughters of an affected male are heterozygous carriers. Number 3. Heterozygous women transmit the mutant allele to 50% of the sons who are affected and to 50% of the daughters who are heterozygous carriers. Number 4. If an affected male marries a heterozygous woman, half their sons will be affected, giving the erroneous impression of male-to-male transmission. Examples of X-linked recessive disorders are Duchenne muscular dystrophy, hemophilia A, X-linked severe combined immune disorder, and some forms of congenital deafness, etc. The blood clotting disorder hemophilia A is one of the several single gene diseases that exhibit an X chromosome linked recessive pattern of inheritance. Males who have a mutant copy of the factor VIII gene will always have hemophilia. In contrast, women are rarely affected by this disease, although they are most often carriers of the mutated gene. Duchenne muscular dystrophy is another example of a single gene disease that exhibits an X chromosome linked recessive inheritance pattern. This condition is associated with mutations in the dystrophin gene. X-linked dominant. Because the gene is located on the X chromosome, there is no transmission from father to son, but there can be transmission from father to daughter. All daughters of an affected male will be affected since the father has only one X chromosome to transmit. Children of an affected woman have a 50% chance of inheriting the X chromosome with the mutant allele. X-linked dominant disorders are clinically manifest when only one copy of the mutant allele is present. Disorders resulting from X-linked dominant inheritance occur far less frequently than other forms of inheritance. As noted, X-linked recessive inheritance can occur, and males are almost always the affected gender, although in very rare cases it is possible for females to acquire two mutant alleles or express milder phenotypes as carriers. With X-linked dominant inheritance, there are no carriers. Expression of the disease occurs in both males and females, and only one mutant allele is required. As might be expected, heterozygous female may be less affected than males because of the presence of a normal, non-mutated allele. The distinguishing feature between an X-linked dominant and an autosomal disorder is that an autosomal mutation is transmitted from males and females to male and female offspring. When a mutation is located on the X chromosome and expressed in a dominant manner, females transmit the mutant allele to both male and female offspring. However, males can only transmit it to females. In addition, affected females may only transmit the mutant allele to 50% of offspring. Males will transmit the mutant allele to 100% of females. Examples of X-linked dominant disorders. The X chromosome linked dominant diseases are rare, but several do exist. For instance, dominant mutations in the phosphate regulating endopeptidase gene, which resides on the X chromosome, are associated with X-linked dominant hypophosphatemic rickets. Similarly, Lett syndrome, a neurodevelopmental disease, is associated with dominant mutations in the methyl CpG binding protein 2 gene. Rett syndrome almost exclusively affects females because male embryos with a dominant mutation in the MECP2 gene rarely survive. Conclusion. From the present paper, it can be said that single gene inheritance is the simplest inheritance pattern. It is also known as unifactorial inheritance. Gregor Mendel first investigated this type of inheritance in his famous studies of garden peas in 1865. The underlying principles of Mendel's work became hallmarks to understanding inheritance. Mechanisms of unifactorial inheritance are often called Mendelian inheritance, and the other mechanisms are referred to as non-Mendelian inheritance. Single factor inheritance in man is the derived conclusion for Mendelian genetics. Although there are various ways of genetic variance in human, the basic model starts from Mendel's one-way transmission, in which the two alleles of a particular organism plays a distinctive role in deciding about the genetic variation and form of the organism. Inheritance patterns trace the transmission of genetically encoded traits, conditions, or diseases to offspring. Thank you.